So when we define metabolism, metabolism really is all chemical reactions that are occurring at any given time in an organism. The organism that we're concerned about is, I hope by now pretty clear, we're talking about a human. So basically, what we're talking about to get metabolism is all chemical reactions that occur at any given time. So it's really the sum total of anabolic and catabolic reactions. So we recognize that cells, they're responsible for their own homeostasis, and cells are continuously breaking down and building up their organelles and their messengers, for lack of a better way to describe products that they may be producing. And as a result, it's important for these cells to maintain what we refer to as a nutrient pool. So they're going to maintain a nutrient pool. And the nutrient pool is monosaccharides, amino acids, and fatty acids. From those building blocks, they can make repairs to various organelles. They can recycle these products when they're breaking down and replacing and regenerating different organelles, and or their products or their messengers, like I mentioned, if it's a cell that makes a hormone or a cell that makes a neurotransmitter or any kind of cytokine, things we've talked about that represent signaling between groups of cells and that sort of thing. So in order to be able to maintain and repair and generate, there's got to be a nutrient pool. And in order for most of those either anabolic or catabolic reactions to occur, we're going to need an energy source. And most of the time, that energy source is going to come from ATP, our high energy molecule that becomes a source of energy when we break the bond between the third phosphate group and the second one to create ADP plus the phosphate group. We get our energy from that broken bond and we create some heat. So we recognize that metabolism is going to be closely tied to thermoregulation. And that's the topic that we'll finish up talking about a little bit later on today. So if we take a look at what we're really speaking about here, I think our next slide gives us a little bit of a picture here. So the beige color, pinky beige color, whatever it looks like to you, is a cell membrane. And within that cell, we can see that we're going to have our lipids and our carbohydrates and our proteins. Okay, and if we follow the carbohydrate pathway first, we see that when we have glucose introduced from food sources, then it's going to undergo a process called glycolysis. That's what this reddish arrow is showing us. And glycolysis uses some ATP and creates a little bit of ATP, but the result of glucose undergoing glycolysis is pyruvate. And pyruvate is the substrate that can enter the mitochondria, especially if we have oxygen available for it. Can you see these things okay? I'm not sure what it looks like for you guys at home. Is it big enough to... Okay, so once pyruvate enters the mitochondria, it's going to be converted to acetyl coenzyme A, enters these two sets of pathways to create lots and lots of ATP. And that ATP is going to be utilized within the cell to perform various functions like repair and growth and the production of hormones or other messengers, et cetera, et cetera, et cetera. So our red pathways here represent catabolic reactions. So we can see that if we happen to be a cell that can store glucose as glycogen, that very first, I'm not sure if this eraser will work here, and it's going to erase everything. So let me just go all the way back. We can see that if we store glucose as glycogen, we're going to have to catabolically break it down to get glucose. So that's what happens there. Now, in a typical cell, we sort of look at this process a little bit differently. Another way to look at this is we have our typical cell here. So we'll draw in our cell membrane, and we'll put together our nutrient pool. So let's put our nutrient pool through the center of our cell, just for a schematic. And we recognize that we're talking about amino acids and fatty acids. And let's just summarize, just to keep it tight, glucose as our primary monosaccharide here. So from the diet, we're going to get some of those nutrients. Also, let's so we'll call this catabolic reactions. So when we're breaking down organelles within the cell to replace them, refresh them, utilize them. So when we talk about maintenance in a cell, when we talk about growth, when we talk about repair, some of those processes result in catabolic reactions, and some of them are going to represent anabolic reactions. Okay, so within a cell, we're going to have catabolic reactions that are going to produce some of those nutrients. And from those nutrients, we have anabolic reactions that are going to be responsible for those processes like repair and growth and so on. You following me so far with this?
Yes.
Yes? No?
Yes.
Yes.
Okay. So the other thing, and the other important role for this nutrient pool to play, is the production of ATP. So we mentioned in that other diagram, we saw glycolysis, and we understand that for most of the ATP production, we have to be within our mitochondria. So this little schematic right here is our mitochondria. All right, somebody's added closed captioning. You know how I feel about that. Useless, but whatever. Okay, now, to take our glucose, for example, and break it down to through the process of glycolysis, we're going to produce a little bit of ATP. But when we add oxygen, we're going to get lots of ATP. So as a result, we're going to produce lots of ATP. And a byproduct of producing lots of ATP is carbon dioxide. So when we look at our little schematic here, we look at our nutrient pool. The nutrient pool is serving two purposes. It's serving for substrates in order for us to make ATP, which is going to allow both of those anabolic and catabolic reactions to occur. The ATP is going to be available to us so that we can do functions like locomotion, in other words, movement, transport. And when we talk about transport, we're talking about exocytosis and endocytosis, for example, and those kinds of cellular functions. So that's why we want that ATP. So this cell is maintaining its own relative homeostasis with respect to this nutrient pool. And when we utilize more nutrients than we can through recycling, we depend on getting new nutrients from our diet. And that was essentially what we talked about with the digestive system, breaking down large molecules of carbohydrates, lipids, and proteins to small enough ones to absorb. When we absorb those, we could reassemble them within the body to make them human carbohydrates, lipids, and proteins, right? So we could eat animal or plant sources and break them down to their building blocks and then reassemble them within cells to make them unique to humans, or at least functional for humans. So our cells are going to continuously absorb carbs, lipids, and proteins from the surrounding extracellular fluid based on digestion and absorption. So what we want to spend today talking about is looking at some of these metabolic reactions, looking at the fate of glucose and certain amino acids and all that sort of thing. So we can take a look at this if we, if I take this picture and just, I want to make this a little bigger. If we take this out and we look at it just in a slightly different way, we can look at what some of the metabolic reactions that are going on in the body look like. So we'll put in our nutrient pool again. We'll put some amino acids in here. We'll put some glucose in here, and we will put our fatty acids. So keep in mind, when we talk about amino acids and fatty acids with respect to ATP production, some fatty acids are gluconeogenic. In other words, they can be utilized for ATP. Not all of them, but some. And that's the same with amino acids. Some amino acids are described as gluconeogenic, which means that they can be used for ATP synthesis, but not all of them. So when we write them in here, it doesn't apply to every single fatty acid or every single amino acid, but it does apply to some. So now what we want to do is we want to look at, we're going to sort of summarize anabolic and catabolic reactions that go on in a cell, typically, in this case, with respect to ATP production. So our fatty acids are going to come from triglycerides. And our glucose from glycogen. And our amino acids from proteins. So what we would do here is that going from fatty acids to triglycerides, what would that be, anabolic or catabolic?
Anabolic.
Right. So that's going to be, those are going to all be represent anabolic reactions. And when we go in the other direction, we're talking about breaking these bigger molecules down to smaller ones. Those represent catabolic reactions. Now we know that when we look at glucose, glucose gets broken down to pyruvate, and that's kind of our favorite way of making ATP, and favorite meaning efficient. It costs us the least. And from here, we can make acetyl coenzyme A, and that means we're inside our mitochondria. And we did that in the presence of oxygen. Now, we can also send pyruvate back the other way in situations where we're going to use another source. or in situations where there's already lots of available ATP, the pyruvate can be sent backwards. In other words, it's a two-way equation. It can go in either direction based on metabolic requirements. Now, when we look at, for example, proteins, those gluconeogenic proteins, we know that in the case of amino acids, some amino acids can be converted to pyruvate, and some can even be converted to acetyl coenzyme A. So we're keeping consistent with catabolic reactions being depicted in our red line. And the same concept applies to some of those fatty acids. Some of them can be converted to pyruvate or acetyl coenzyme A, and in those cases, they're being utilized then for ATP production. Now, we can go in the reverse direction. So we can take acetyl coenzyme A and reassemble fatty acids, but not pyruvate. Typically, that's a one-way pathway. And again, when we look at amino acids, we can take pyruvate and reassemble to amino acids, but not if the amino acid has already been broken all the way down to acetyl coenzyme A. So we're looking at anabolic versus catabolic, and we can further identify some of these reactions. So for example, when we break down glucose to pyruvate, we call that what? What metabolic reaction is that called?
Glycolysis?
Right. Glycolysis. Okay. So we got a couple other things to be aware of. So if we look at number 2, if we call this one here number 2, number 2 in this case is going to be called what? So it's catabolic.
Glycolysis?
Glycogen olysis, breaking down the glycogen, right? Because it's catabolic. Back to glucose. And that would then, we would go in the opposite direction. Glycogenesis. Okay. And that leaves us with gluconeogenesis. Gluconeogenesis is the conversion to a glucose molecule or one of its substrates, pyruvate or acetyl coenzyme A. And so we have that happening under these circumstances here. So this would be a number 4, and this would be a number 4. So we're looking at gluconeogenesis by definition is by literal definition is making glucose from something other than something new than a carbohydrate. So we're talking about making it from some of our amino acids in this pathway or some of our fatty acids in this pathway. And you can see that gluconeogenesis in physiological terms doesn't necessarily mean you're going to create the molecule glucose. In a lot of cases, you're actually making pyruvate or acetyl coenzyme A. But because they are being introduced to the pathway to make ATP, that's referred to then as gluconeogenesis, because it's going to contribute to ATP production. Okay, now, before we leave this page, the other terms that I want you to be aware of, because they're going to be utilized throughout the rest of the lecture today, is we want to be aware of a condition known as the absorptive state and the post-absorptive state. And the reason you want to be aware of these terms is that the direction of action, whether we're going anabolic or catabolic, will change based on the state of the body at a given time. So an absorptive state is during or immediately after a meal. So sometimes the body is considered to be sated. And later on, when we look at regulation we'll see in the hypothalamus, there are neurons that are described as being the satiety center, the center for being full. And the fancy word for that is being sated, or it's a satiety. It's a funny word, center. And post-absorptive state is the state of fasting. So this is between meals. So you can appreciate that when we're fasting, the demand for ATP is going to be higher because our blood glucose levels are going to be lower. So if our blood glucoses are lower, we're looking for alternate pathways, additional pathways to create ATP. When we're in our absorptive state immediately after a meal, by definition, our blood glucose levels have been satisfied. And when the blood glucose levels are satisfied, that means there's enough glucose in the circulation for ATP production. So instead, what we would see is more of the anabolic pathways during the absorptive state. Does that make sense? Everybody following me with that so far?
Yes, sir.
Okay, so where does all this stuff happen? Everybody will love seeing this picture again. So we know that absorption of nutrients is going to be the responsibility of, well, about 90% of it is the responsibility of the jejunum in the small intestine. And some of the features in the small intestine that favored this absorption was about surface area. So we had villi. That's what we're looking at here. And if you look really closely, I don't know how well it shows up for you, there are the microvilli shown on the so-called apical surface of those columnar cells on those villi. And within the villus, we can see the circulatory system. We've got a venous system, and we know that the venous system is going to finish on what? Where will these veins drain into?
Vena cava, inferior vena cava.
Before that?
The
liver.
The
Yeah,
portal.
through what mechanism? What mechanism?
portal vein,
Which kind of portal vein?
the hepatic portal vein.
Yeah, we got to put that in. There's more than one.
Yeah.
Hepatic portal vein, or system, if you called it that, that works. Okay, and that's what's responsible for giving us our nutrient-rich venous blood delivered to the liver. Okay, and this greenish structure right here, what does that represent?
Lacteal.
Yeah, that's our lacteal. And our lacteal is a lymph vessel. And in this case, this lymph vessel is serving a purpose for the digestive system. It's going to absorb long-chain fatty acids in the form of something called a chylomicron. And the chylomicron is going to be collected up. The chylomicrons, large chain fatty acids, are going to be collected up by these lacteals and delivered to the circulatory system through the thoracic duct. And the thoracic duct emptied into the subclavian vein. And the subclavian vein would end up in the vena cava. So we're returning the large chain fatty acids to the circulatory system, but we're bypassing the liver. So some of those lipids, small chain fatty acids, are going to be carried the same way as amino acids and monosaccharides are through the hepatic portal system, but the larger ones bypass it. So from the liver, like somebody mentioned, we are going to empty into the vena cava from the liver and enter the rest of the systemic circulation that way. So we want to look at absorption of these molecules too, and what mechanisms are going to be utilized. So the summary mechanism that's going to be utilized for absorption is going to be active transport, both primary and secondary active transport. There's going to be some diffusion, so passive transport. There's going to be some facilitated diffusion, so we need some unique channels for that. And we're even going to see some exocytosis of some of these molecules in order to satisfy absorption and introduction to the system. Because remember, if the food stays out here in the lumen, it's of no use, right? We have to absorb those nutrients. So we have to be successful at digestion. starting in the mouth with chewing, and then continuing on in the stomach with exposure to acids and so on, and then enzymes in the small intestine to get those large food molecules small enough to be absorbed. And then we can send them mostly to the liver, where the hepatocytes are going to determine the state of the body, absorptive or post-absorptive, and then determine where we're going to go from there. Are you guys can still see the screen, right?
Yeah.
Yeah, because I just had some stuff flash up on me here. I don't know. Sometimes we could get cut off and I won't know. I could be sitting here talking to myself. So we're going to come back to that when we look at specific substrates here. But here's our absorption summary. And this is giving us an idea here. So here's our chylomicrons. Those are the lipoprotein formation from long chain fatty acids being carried in a protein within a protein molecule so that they can be delivered to the circulatory system up here at the left subclavian vein in this diagram here. So you can follow the green lymph line all the way to reintroduction, but we bypass the liver. And the hepatic portal system is picking up the amino acids and the monosaccharides and the smaller chain fatty acids for delivery to the liver and metabolic fate being determined there at the liver. So that's our recurring theme for every nutrient that we're going to be looking at today. So we'll take the main organic compounds and look at the fate or the metabolism of each of those as a separate entity. So we're going to start with carbohydrates. They're the ones we probably are most comfortable with, the ones we know most about. So we know that the digestion of carbohydrates starts in the mouth. And the goal of digestion is to break down carbohydrates like pasta and potato and vegetables and fruits and so on, breads, basically to their building blocks, monosaccharides. And the monosaccharides that are of some use to humans are glucose, galactose, and fructose. So in other words, these are the monosaccharides that we have enzymes for to be able to break them down and then to be able to utilize them. And this represents, our favorite way of making ATP, because in the liver, galactose and fructose, they can each be converted to glucose. And glucose is our easy way of making ATP, our least expensive way for making ATP. So glucose is our preferred source for ATP production. And the utilization of glucose is going to be dependent on the cell's requirement for ATP. So the busier the cell, the more ATP, the more glucose we're going to be looking for. So when the need for ATP is high, glucose is going to be utilized. And that process of undergoing glycolysis and then entering into the mitochondria is sometimes referred to as oxidization. We've oxidized the glucose. When the need is low, the glucose is going to be stored. We're going to reverse our pathways, and we're going to store glucose in the liver, for example, as glycogen. And the other major tissue group that can store glycogen is which one?
Liver,
Liver is one. And what's the other one?
muscle,
Yeah, muscle. Specifically, skeletal muscle and cardiac muscle. But the difference is when the liver stores glycogen and we're in a post-absorptive state, that glycogen can be broken down to glucose and shared systemically. When muscle tissue breaks down glycogen, it's for its own utilization. So typically the glucose that's being broken down in muscle tissue is not released out of the muscle tissue. It's going to be utilized for muscle cell function. And the demand for ATP in muscles is very high. We know from what we remember way back in semester 1 that muscle contraction requires ATP and muscle relaxation also requires ATP. So there's a high demand for ATP, really, no matter what state our muscle tissue is in. And that applies to cardiac muscle as well, which we know has to be able to contract and relax in a cycle about 60 to 100 times in a minute, every minute of the day, or else we're going to notice a problem. So when our glycogen levels get maxed out in the liver is going to convert the rest of any glucose that might be available to fatty acids. So the glucose can be converted to glycerol and fatty acids to create triglycerides. And some of our cells, we know, because we've been talking about it, can convert glucose from some of our amino acids. So, and when we convert, if we look at this, if we look at if we look at the fate of glucose, so if we take our glucose and we sort of summarize what I've just been saying, so we know that glucose is going to undergo some steps. We're not going to worry about those steps, but there's a few steps where glucose is acted upon by some enzymes that ultimately result in the formation of pyruvate. Now, I'm just going to put in that step. I don't want you to worry about what it is that is involved in that precursor before pyruvate, but recognize that under certain conditions, basically when the demand for ATP is low, then this substrate that's in this step right here can be converted to glycerol. And if we add fatty acids to glycerol, we can get monoglycerides, diglycerides, or triglycerides, but we tend to prefer triglycerides to store potential ATP because, again, these steps are reversible as well. But the triglycerides are going to get stored in adipose tissue. So we'll come back to that step in just a minute. So the pyruvate, we know, is going to be able to enter the mitochondria, where it gets converted to acetyl coenzyme A, in the presence of oxygen, right? We're all familiar with this. We've used this many, many times. And the triglycerides, the glycerols plus the fatty acids, so to sort of complete that pathway, the fatty acids can be converted somewhat easily, and this is a reversible step, to this acetyl coenzyme A. Some of our amino acids can be converted preferentially to pyruvate under these conditions here. And again, we can use our steps here. This process was called glycolysis, right? If we let's go back to yellow. So in this picture here, that's glycogenesis. This is glycolysis. If we follow fatty acids to acetyl coenzyme A, okay, we better try to stay, I think I'm staying consistent with the numbers we used on the previous slide, but as long as we've got these marked, that one would be called what? Number 3?
Glycogenolysis.
Yeah. And so number 4 was gluconeogenesis, which is anytime we're going to make one of these substrates for ATP production from something other than a carbohydrate. So when we look at this little schematic here, never mind all the green stuff, what we're looking at is this really represents the metabolism of carbohydrates. When we build up glucose to something new, glycogen, that's anabolic. When we break down glucose to pyruvate or to glycerol, that's catabolic. When we add glycerol plus fatty acids to create triglycerides, that particular reaction is anabolic. But when we take the triglycerides and we convert it to fatty acids to acetyl coenzyme A, we're going catabolic again. You follow me there for anabolic and catabolic pathways?
Can you please
Yes?
repeat
Yeah.
again?
Okay. So if we're taking glucose and making it into a bigger molecule, glycogen, that would be an anabolic reaction. If we're taking glucose and making it into a smaller molecule like pyruvate, that's catabolic. So glycolysis, it's got that word lysis in it. That's a pretty big hint that it's breaking something up, right? So that's a catabolic reaction. Glycogenolysis, up there, number 3, is another example of a catabolic reaction. We're taking glycogen and breaking it down to its building blocks, glucose, right? So glycogenesis is anabolic. Glycogenolysis is catabolic. Does that make sense?
Yes, thank you.
Yeah. Everybody else okay with that?
So glycolysis is also catabolic?
Yep, because we're taking glucose and making it into pyruvate. So you might remember, we didn't go into a lot of detail in the biochemistry stuff, but glucose is made up of 6 carbons in a ring, right? And pyruvate is 2 molecules of 3 carbons. So we took the 6 and made it into 2 molecules of 3 each. And that's maybe more than we need to know now, but you can appreciate that that's a breakdown, right? That's 2 smaller molecules. Does that work?
Yeah.
Okay. Now I mentioned that we can also take fructose and galactose and convert those. Again, the hepatocytes can do this. They'll take the galactose from our diet and convert it to glucose in the liver. And fructose can be converted to glucose in the liver too. The problem is what we're recognizing more and more and more with fructose is that when our diets are high in sucrose, some of you might remember from way, way back, sucrose is a disaccharide made from glucose and fructose. So diets that are high in sucrose, like our fast foods and our simple sugars and our stuff that's been fortified, so-called fortified, with corn syrup and things like that to make them taste really sweet, can overwhelm the hepatocytes that are trying to convert the fructose to glucose. So what can happen is it's going to be metabolized by hepatocytes, and it can be converted to glucose, but it also produces some uric acid. And if we produce large amounts of uric acid, what happens to pH?
Increase.
pH is going to
go down.
It's going to decrease. So acidity increases, but that means pH is going to drop. So uric acid accumulations because of excessive fructose metabolism can be a concern if it's chronic and if it's excessive. We also know that we can overload pyruvate. We can make too much pyruvate through this pathway, and that's going to overload the mitochondria. And as a result, what we're going to do is send the pathways this way. So we're going to start to make excessive amounts of triglycerides because the mitochondria has plenty of pyruvate. So that's going to redirect this pathway in this direction. And when we produce triglycerides because of excessive amounts of fructose, not all of it is going to get deposited in adipose tissue. Some of the excess triglyceride are deposited in the liver instead. And then we get what's called a fatty liver. Now, we know, some of you may be aware, that in chronic exposure to alcohol, the liver becomes fatty. So this is described as being non-alcoholic fatty liver. And it's due to this excessive amount of fructose metabolism. So, and as a result of fat accumulating in the liver, we're going to lose some of the functions of the hepatocytes in that liver. So it becomes a problem. It becomes a dysfunctional liver. And we know from what we talked about a couple of weeks ago, liver is responsible for all kinds of things, not the least of which is this metabolism for glucose and the determination of whether we're going to go glucose. Remember, we had 3 fates for glucose: blood glucose, glycogen, and triglycerides. In the North American diet, because we tend to be relatively high in our carbohydrate ingestion, generally, that most, probably typically, around 50% or so of our carbohydrates that we take in become triglycerides and stored in adipose tissue. Now, if we go higher than that, if our fructose intake is so high that it overwhelms the system, then we start to see the deposits of triglycerides in other tissues than adipose tissue. And that becomes a pathology. You could see deposition in the heart, for example, or the kidneys, or in blood vessels, and increases the risk for things like atherosclerosis, which you're going to talk more about when you get into pathologies and so on. So the issue with fructose and those simple sugar diets, you know, and the emphasis on fructose high or corn syrup type sweeteners is this overwhelming of our hepatic pathways, resulting in accumulation of products that we're not interested in, that are not going to be helpful, like that uric acid. We know that when uric acid, another thing just to keep in mind, not something I'm going to ask you a question on, but when uric acid levels accumulate because of this fructose metabolism drive, it also inhibits something called nitrous oxide. Some of you might know nitrous oxide. We'll talk a little bit about it when we get to the kidney as well. Is a promoter it's a vasodilator. So when a vasodilator affects blood vessels, it's going to reduce blood pressure. So it's one of the mechanisms we take advantage of to regulate blood pressure. If we reduce the amount of available nitrous oxide, then we have the risk of high blood pressure. So chronic exposure to this fructose is going to have us depositing triglycerides in tissues we'd rather not have them deposited in. It's going to overload the mitochondria and force that redirection of glucose breakdown, glycolysis, to the triglyceride pathway. And it's going to raise blood pressure. So it creates a series of events and factors that are promoting pathology, right? So that's one of the reasons why, from a dietary perspective, we should be focusing on complicated or complex carbohydrates that are less easily broken down to pyruvate without some time, right? So if we have to pull them from complexes rather than from simple carbohydrates. You following me? Am I making that clear enough?
Yes, sir.
Complex versus simple, that's really what that boils down to. Okay, so when we're looking at carbohydrate metabolism, what we're looking at is those four pathways: the glycolysis, the glycogenesis, the glycogenolysis, and the gluconeogenesis. And we can summarize all of that if we take a look at our systems, so when we're looking at carbohydrate metabolism, we're going to look at an epithelial cell. So this is an epithelial cell. These are the villi. Okay. And we know that our epithelial cell, if it's successful in absorption, it's going to drain through veins and end up being delivered to the liver. There's our two lobes of the liver. And from the liver, will drain into the heart. And from the heart, we're going to deliver to all the cells and tissues in the body, including and we're going to make this our typical cell. So we're going to receive, in this case, our glucose through the blood glucose delivery pathway. So what we've got now is, if we go back now and look at glucose, galactose, and fructose, those are the monosaccharides we're interested in the carbohydrate metabolism. So glucose has to be entered into the small intestine enterocyte, right? The site that's the cell that's responsible for absorption. And that's going to be through a mechanism that we call secondary active transport. So these receptors, we should all be comfortable with the concept of receptors now. For glucose and galactose, that process is facilitated by receptors that are known as SLGT1 receptors, or promoters of absorption in this case. The S is for sodium, GL for glucose, transporter type 1 is responsible for secondary active transport of glucose and galactose into the cell. Fructose enters through a glucose transporter that's called glucose transporter number 5. That's unique to fructose. That's the way these monosaccharides are going to enter the digestive cells in the small intestine, and then they leave through, all of them leave through a GLUT2 receptor. So that's how we're getting our monosaccharides introduced to the hepatic portal system, right? So this represents our hepatic portal system. When the hepatic portal system delivers, in this case, our monosaccharides, we're going to be able to convert galactose and fructose to glucose. We're going to reestablish blood glucose levels. If our body is easily reestablished, if we easily reestablish blood glucose levels, it's because we're in that absorptive state. Then the primary direction of metabolism is going to be to the storage forms, glycogen, and then triglycerides. If the blood glucose levels are low because we're in a post-absorptive state, then we're going to see catabolic reactions. The triglyceride pathway is going to be reversed, or we're going to take the fatty acids and make them into pyruvate or acetyl coenzyme A. We're going to take amino acids and make them into pyruvate. We're going to take our monosaccharides and make them into pyruvate, and glycogen to glucose and pyruvate to make ATP, and release all of that glucose that's being produced in the liver into the circulatory system for delivery. So we're going to follow our glucose through the circulatory system, contribute to the nutrient pool for our cells. This represents our cells, systemic, right? We're going to send a little bit here for the cells of the requirement for the cells themselves, right? This would be our hepatic artery, right? And this was our hepatic portal So we talked about nutrient-rich venous supply, oxygen-rich arterial supply, and our blood supply here, blood in this direction here, is to supply nutrients to those cells. They need their own set of nutrients for survival. Right? Are we following this? Does this make sense? I don't know if this is helpful or not.
It's helpful.
Okay, so in the absorptive state, we're going to see mostly anabolic reactions to glucose dominate. So that means we're going to produce glycogen and really lipogenesis, right? And in the post-absorptive, glycolysis and gluconeogenesis are going to dominate. Because what we're looking for here is there's a demand now for ATP. There's a demand for glucose based on the fact that we haven't got fresh nutrients being delivered to the cells. So we're in that fasting phase. We're between meals, right? So that's what we would expect to see with respect to the carbohydrate pathways driven primarily by the liver. Does that make sense? We okay with that? No? Yes? Kind of?
Yeah.
Okay. Because so now what we want to do is we're going to move on and do the same thing, talking about the lipids, and then we'll do it again with the proteins. So if we look at lipid metabolism, so again, starts with digestion and absorption, right? And we know that lipids are a primary source for ATP production, but also play a much bigger role than carbs do in respect to structure and function within the body, starting with the cell membrane, where 80 to 85% of the cell membrane is from phospholipids, another significant contribution by cholesterol. And then we have our steroid-based hormones and so on and so on. So they play a much bigger role in structure and function in the body, but still maintain a very important role in the production of ATP, being available for the production of ATP. So when the demand for ATP from lipids is low, the fatty acids are going to be stored as triglycerides. And some are going to be available for the anabolism, for example, to produce or repair cell membranes and so on. And the triglycerides are produced in the liver and stored in the adipose tissue. So remember earlier I mentioned that some fatty acids are described as being essential. Well, I mean, not later, I didn't say that earlier today, but when we use that word essential, when we're talking about nutrition, what is that word telling us? If we say that there are some fatty acids that are essential, what does that mean?
You need it from the diet.
That's right, because we can't synthesize them. We don't have the enzymes or the ability to synthesize these fatty acids that are described as being essential. So vitamins and minerals are described as being essential. Some fatty acids are described as being essential. Some amino acids are described as being essential. And the term in this case means that we require them from an outside source. So that means our diet. We need to get those things from our diet. So again, I mentioned that some, because I'm telling you by saying it that way, that I don't expect you to memorize them. It's not important for us, but it is important just to understand the concept, right? So when we look at catabolism of lipids, what we're saying is we're taking triglycerides, that was stored in adipose tissue, and we're going to break it down to its building blocks. Glycerol, which we know from what we looked at on the previous slide, was derived from glucose, and fatty acids. And this process is otherwise described as lipolysis. So if we have a demand for ATP and the demand is relatively low, then this becomes a preferred pathway for ATP production because it allows us to spare glucose. So there's lots of scenarios in the body where we want to spare glucose for more fight or flight type responses, for more emergent ATP production. So when the ATP demand is relatively low, we can mobilize these triglycerides through lipolysis and make them available for low level or low intensity ATP demands. So think of jogging versus sprinting. In a well-trained individual, you can spare some of that glucose while running if you run at a slower, more steady pace. And that allows you to spare or save the glucose for a sprint. When you look at these well-trained marathon runners, for the first 20 or 22 miles of their race, they're primarily using a triglyceride pathway for their ATP source. Now, I say primarily because it's never on and off, right? It's going to be a shared mechanism. But when you can utilize the triglycerides and their pathway for ATP production, you save some of the glucose for when you need it quicker. Glucose is a faster pathway. So the glycerol can be converted back to glucose, or it can enter the pathway that was glycolysis and become pyruvate. In either case, being able to be utilized for ATP production as well. The fatty acids can be converted to acetyl coenzyme A, like we showed in that diagram on the previous slide. And sometimes if there's excessive amounts of fatty acids being utilized, we get an accumulation of something called ketone bodies or keto acids. So these can be utilized that way. And if there's excessive fatty acids available for ATP production, so excessive meaning more than what we can use, we accumulate some of the acetyl coenzyme A is converted to ketone bodies or keto acids. And again, you see that word acid, so what's going to happen to pH?
Decrease.
Right, we're going to get a decrease in pH. We're going to enter into potentially acidosis. And acidosis is a concern because lower pH is going to have a direct effect on protein structure. We know if we alter protein structure, we're going to change function. And oftentimes that's irreversible. So that becomes a problem that we have to deal with, only if it's being excessively utilized. So normally, under normal conditions, this is not a problem. The keto acids can also be cleared through the liver or actually reused as a source of ATP. So there's lots of normal utilizations for these keto acids. But in pathologies like type 1 diabetes, this is the pathway that becomes favored because glucose is not available in someone with type 1 diabetes. So these keto acids accumulate under those circumstances, create a condition called ketoacidosis, which becomes life-threatening. So again, most of the time, unless we're dealing with a pathology, this pathway is not a major concern. In fact, even our neurons can use some of these ketones as a source of ATP when under most conditions they can only use glucose. So it's not a total disadvantage that way. Now, I'm going to squeeze in over here, anabolism.
Sorry, what was the word that you put right before 4-ATP?
Utilized. Is that what you're looking at
Yeah, okay, thank you.
here? After all this time, you're having trouble reading my writing. I can't imagine. You know, I'm kidding. I have terrible writing. I get that. And it isn't because I'm left-handed. It's more than just that. So hopefully these capital blocks you can read a little easier. So for anabolic pathways in lipid metabolism, when the ATP demand is low, carbohydrates, lipids, and proteins are all can be stored as triglycerides, right? So we're going to find out that proteins can't be stored in the body at all as a protein. So they're going to get converted to triglycerides. We already know the pathway that results in excess carbohydrate being stored as triglycerides, and the same goes for lipids themselves. They're going to become triglycerides when ATP demand is low. We store. So in anabolism, we're taking the fatty acids and we're adding them to glycerol, and we get triglycerides. Now we can get monoglycerides and we can get diglycerides, but our typically stored form of fatty acids is going to be as triglycerides. So, and in either case, we're building onto the glycerol molecule, so they all represent anabolic reactions. Okay, so that's basically the anabolic pathway that we're talking about when we look at what happens to fatty acids when they're absorbed into the system. So they're going to be anabolized to triglycerides. The other thing that they can be turned into within the liver are lipoproteins. And chylomicron is an example of a lipoprotein, but that's occurring in the small intestine cells. In the liver, we get very low density lipoproteins, low density lipoproteins, and high density lipoproteins. So these are molecules that carry cholesterol. In the case of very low density lipoproteins, triglycerides, but low density lipoproteins carry cholesterol from the liver and deliver it to all the different cells in the body that require it. And HDLs take excess cholesterol in the periphery and return it to the liver, either to be recycled or to be redistributed through LDLs. So these are carriers of cholesterol. In the case of very low density lipoproteins, some triglycerides, just like our chylomicrons were. So that's what a lipoprotein is. It's a protein molecule that allows us to carry, in this case, mostly triglycerides or cholesterol in the blood to their various targets that we're looking for as far as that goes. And again, what we'll do is add a layer in here. And we'll look at what happens to our triglycerides. So if we start with triglycerides, and we had our triglycerides were formed from fatty acids and glycerol. So glycerol plus fatty acids is going to be a anabolic pathway, breaking down triglycerides to glycerol and their fatty acids would be a catabolic reaction. The glycerol came from glucose being converted to pyruvate. So that was a two-way pathway. And our fatty acids, pyruvate ends up being acetyl coenzyme A, and acetyl coenzyme A can be converted to fatty acids as the reverse is true. We make lots of ATP in our carbohydrate pathway, and our fatty acids are going to be converted in the liver to products like cholesterol and phospholipids. Or like we said in that previous lipoproteins. So if we look at the breakdown of triglycerides to fatty acids, let's put a color code in here. Number one is lipolysis. So when we follow our pathways that result in the synthesis of triglycerides, so this would be here. Let me get rid of this. Going in the wrong direction. So we're lipogenesis when we're heading towards the triglycerides, or if we're synthesizing additional lipid molecules. These would all be examples of lipogenesis. When we send acetyl-CoA towards fatty acids, lipogenesis. When the glycerol molecule is being produced in order to be able to form triglycerides, that would be a form of contributing to lipogenesis, so anabolic. And then lipolysis is the breakdown going in the other direction. So this is describing our lipid metabolism. And if we try to summarize that with our main tissue groups, here's our enterocyte in the small intestine. Here's our hepatic portal pathway, our bilobed liver, draining into the heart, delivering okay, with one major difference, because now we're looking at lipid metabolism, we have to introduce the lacteals. So the lacteal is going to be drawn in like that. So our chylomicrons okay, and chylomicrons, they actually undergo a process of not sure how best to draw this out, but basically exocytosis. So our fatty acids from our diet, they're going to enter through the circulatory, through the digestive tract. So this represents the lumen. So fatty acids, they enter the small intestinal cells because they're acids, because they're lipids, they don't need a channel or a transporter of any kind. They're going to be able to go through what we call, anybody remember what we call? Fatty acids can cross the cell membrane through what process? Because they're lipid soluble.
Diffusion.
Yeah, I think somebody's crazily typing. I can hear typing. Simple. diffusion. So we can go through simple diffusion, and what's the other type of diffusion? Not simple, but
Channel mediated?
sorry, what mediated?
Channels?
Channel, yeah, you're right. And it's actually referred to as facilitated diffusion, and the facilitator is the channel. So you're absolutely right as far as that goes. But in the case of lipids, we don't even require channels. We can go through simple diffusion. So our short chain fatty acids and our long chain fatty acids can be absorbed through the villi and microvilli pathway. Enzymes, generally referred to as lipases, will allow that simple diffusion to occur. Some of those longer chained fatty acids are going to be combined into chylomicrons, and the chylomicrons are going to be released from the small intestinal cells through exocytosis, picked up by the lacteals, and delivered to the circulatory system through the thoracic duct, bypassing the liver. The smaller chain fatty acids are going to follow the same pattern as we described with the carbohydrates, with glucose. And fatty acids that are going to be released into the circulatory system will be pumped through and delivered to cells for utilization as part of their nutrient pool. So everything else is the same with this difference. So that's a difference for when we talk about the metabolism of lipids. They're going to have a component of the lymphatic system introduced there as part of that process. Okay, do we need a little break before we talk about proteins?
Yes, please.
Okay, we're on resume. So when we talk about the digestion of proteins, our goal for the digestive system is to break them down to the building blocks, otherwise known as amino acids. Some amino acids are described as being gluconeogenic, which means they can be utilized for ATP. Most amino acids are going to be synthesized into new proteins that are important for structure. For example, structure or functions in the body, protein-based hormones, enzymes, molecules that we know of like hemoglobin and myoglobin, antibodies, plasma proteins that are synthesized in the liver fall into categories like fibrinogen and angiotensinogen. We know that some proteins are contractile in nature, myosin and actin, structural in nature, collagen and elastin. So these represent reassembly of amino acids that came from our diet into proteins that are specifically functional for humans. Right? So we take our proteins, we break them down to amino acids. Some can be used for ATP and the others for structure and function. And I'm not going to go over all that list again, but basically we're talking about, for example, collagen and elastin as functional or structural, enzymes and hormones. And then in between, we've got channels and pores and receptors, good old receptors, et cetera, et cetera, et cetera. So these are all going to represent different sort of fates for proteins in the body. Now, we can't store proteins. So as we've mentioned before, so excess amino acids are going to be converted to fatty acids and stored as triglycerides. Whoops. So We are unable to store proteins. We can store glucose as glycogen. We can store excess lipids as adipose tissue, triglycerides as adipose tissue, but proteins are going to be converted to triglycerides. And so we don't have a stored pool of proteins the way we do glycogen or adipose tissue. There are some amino acids in the cellular pool, but not stored as some sort of potential reserve when we get into fasting situations. So when we look at protein catabolism, that was our breakdown. And for a major driver of protein catabolism that we've talked about is cortisol. And we also some other hormones that can promote protein catabolism would be epinephrine or norepinephrine, but a major one, we know that part of the problem with cortisol being our so-called stress hormone is that prolonged exposure to stress, elevated levels of cortisol is eventually going to lead to atrophy, especially noticeable in our protein based tissues like muscle tissue and bone tissue. So it leads to atrophy under those conditions. So basically what we have when we talk about protein breakdown is we've got some cells that have a natural cycle that results in some protein breakdown. We know red blood cells are going to break down. They have a defined lifetime. And part of the breakdown includes the breaking up of the hemoglobin molecule. Remember, there were 4 chains of proteins associated with that hemoglobin molecule. So part of the recycling of red blood cells includes the breakdown of hemoglobin to the amino acids, and then those amino acids can be reassembled to make new proteins or even new hemoglobin, as far as that goes. So in the liver, protein breakdown converts some amino acids to fatty acids, like we mentioned. Some amino acids will be converted to ketone bodies, ketone bodies. And some can be converted to glucose. And that process was called gluconeogenesis. So these amino acids that can be utilized for ATP, because they can be converted to glucose, that's going to all be the responsibility of these hepatocytes, determining where our needs are, right, based on the demand for ATP and so on. So when we look at protein anabolism, now we're talking about building up. We're taking peptide bonds and creating amino acid chains. And that's going to be driven by ribosomes under the direction of messenger RNA located in endoplasmic reticulum, specifically rough endoplasmic reticulum when we're synthesizing proteins, right? Driven by our DNA signaling, our genes that are responsible for assembling proteins through messenger RNA and ribosomes and endoplasmic reticulum. And almost all cells are going to be synthesizing some amount of proteins in this way. So that type of protein anabolism is going to be stimulated by IGFs. Remember IGFs? They were released from the liver in response to the liver being targeted by what? Growth hormone, thyroid hormones, insulin, testosterone, and estrogen. All of these hormones promote protein anabolism. right? And proteins of illness or the need for repair. So too much dietary protein does not increase amino acid anabolism unless it's combined with vigorous weight training and or resistance exercises. So in other words, like I mentioned before, we can't store excessive amino acids as a potential source for protein anabolism in times when we might need it. Instead, it's going to be the fate of amino acids are going to be either gluconeogenic or fatty acid or actually anabolized into new proteins. So adding amino acids to your diet in the interest of creating a nutrient pool of extra amino acids doesn't work. And the only time extra dietary protein is going to be a benefit is if you also combine it with vigorous resistance or loading type physical activity. In that way, you can take the benefit of extra protein. But if you're just talking about anabolic and catabolic reactions that are occurring at rest, bringing in extra protein doesn't have a great advantage. You have to combine it with that added resistance type loading on muscle tissue to get the advantage there. So 9 of our 20 known amino acids in nature are known to be essential. So we have to get those from our diet. We can make the other 11 from those original 9. Okay, so again, using our little schematic for proteins, at the risk of being a bit repetitive, hopefully nobody minds the repetition, because repetition is good for us, right? Hepatic portal pathway to the liver, 2 lobes of the liver, draining from the liver to the heart, systemic circulation, cells and tissues of the body, right? And so what we're looking at down here is that we're going to have some short chain peptides and amino acids. And amino acids are able to be delivered through this system by what we call peptide transporters. So peptide transporter type 1, they utilize secondary active transport for some of our dipeptides and tripeptides. That would be our short chain amino acid groups, right? And then individual amino acids can go through a secondary transport mechanism or an active transport mechanism through usually using sodium, like we talked about when we talked about glucose transporters, those sodium glucose transporters, same idea for amino acids, individual amino acids. And then they are going to just be able to go through simple diffusion into the hepatic portal system. When they've been processed through the small intestine, simple diffusion from the intestinal cells into the hepatic portal system to the liver. And then the liver is going to determine whether the fate of those amino acids is to be released into the circulation for anabolic reactions to occur systemically, or anabolic reactions occurring within the liver, or catabolic reactions occurring within the liver, right? And that again was metabolism or homeostasis of proteins that we talked about when we talked about functions of the liver. So again, we're going to go through, we're going to be utilized in the liver and then into the circulatory system for delivery, nutrient pool, And we've got our sort of pathway there for the proteins that were digested and then absorbed in our system in that respect as well. We following this okay? We all right?
Yeah.
Okay, so if we want to sort of summarize what's going on then in the body, is here is our picture of a summarizing of everything that's going on. Here's our amino acids, here's our glucose, here's our triglycerides being absorbed from the digestive tract, 90% through the jejunum. Absorption in the liver, the glucose is going to be determined to be either released into the circulation when the demand for ATP is satisfied. We can see it go in different directions, into either stored as glycogen or stored as fatty acids. So in this case, we're talking about our absorptive phase. So in our absorptive state, we're storing, we're favoring mechanisms that result in the storage of these nutrients, of these organic compounds. So we're taking the excess glucose and storing it as triglycerides. We're taking triglycerides and storing in adipose tissue. So if we are chronically in a resting state or chronically full because we eat a lot, then these pathways are going to dominate. And as a result, we're going to accumulate triglycerides. We're going to accumulate adipose tissue. In North America, this is at the root of many of our chronic pathologies: cardiovascular disease, obesity, type 2 diabetes, is because we eat too much. And it's not just that, but that's a factor that comes into play. And we mentioned what happens with excess fructose from, high intake of simple sugars, of corn syrup based foods, which is basically what most of our processed sugar sources are: chocolate bars and anything that's been sweetened industrially. So this is where we're satisfying the ATP requirements, and so as a result, we can store in other mechanisms. So here's glycogen here, and here's glycogen here. Right, here's the formation of fatty acids that are ultimately going to be stored in our adipose tissue. Now, if we look at what's happening in our post-absorptive situation, so between meals, fasting, when we are not meeting the demand for ATP, then what we're going to see is a reversal. We're going to see a dominant situation where the glucose, so here's glycogen, glucose pathway is heading into the circulatory system. And amino acids are going to be converted to glucose, and fatty acids are going to be introduced to the glucose pathway so that we satisfy the demand for ATP in the brain, central nervous system. We see mobilization of triglycerides from adipose tissue into the circulatory system and then to the liver. And it's all about now mechanisms that are going to be available to produce ATP. Does that make sense? Do we see that okay? Yes, no, maybe?
Yes.
Okay, so what about regulation of all this stuff? So regulation of metabolism, maybe even more simply recognized as the regulation for eating, is going to be the responsibility of what group of neurons in the central nervous system? Where do you think the neurons are that regulate hunger?
Hypothalamus.
Yes. It's not like we haven't talked about that before. So basically what we have is we have a group of neurons that are responsible for signaling when you're full. And that's that word I used earlier today. So these neurons are responsible for telling us we're full. They're described as being the satiety center, satiety center, but satiety is the way it's typically pronounced. And the opposite of satiety is hunger. So we'll call that our hunger center. So basically all it is a group of neurons that will be excited when you're hungry or excited when you're full. And in either way, the message then from the efferent pathway from the hypothalamus will depend on which center is being activated. So some of us will remember that there's a hormone produced in the stomach that stimulates the hypothalamus to tell you that you're hungry. Anybody remember what that one was called?
Ghrelin.
Yeah. So ghrelin is described as a hormone. It's stimulatory to the hunger center. So it was gastric, right? Cells in the stomach were responsible for producing ghrelin. And when it gets released into the circulatory system, it targets the hunger center to signal that you got to eat, you got to do something. Now, another signal that's pretty straightforward, if we think about blood glucose levels, what would the blood glucose levels be at if it was going to be stimulatory to the hunger center? Would they be low or would they be high?
Low.
Yeah. So that means that we can put the opposite one up here because it's pretty self-explanatory. If we have high blood glucose levels, that's going to be excitatory to the full center, right? The center that says you've got plenty, you don't need to eat anymore. Now, what hormone that we've mentioned and haven't spent a lot of time talking about, but there's another hormone that gets released to indicate to the hypothalamus that we're full. Anybody remember what it's called?
Leptin?
Yeah, excellent. And it's produced where?
Adipose tissues?
Yeah. So leptin's produced by adipose tissue, and really what it's being released in response to is depositing triglycerides. The more triglycerides that are being deposited into adipose tissue, the more the signal is that we've got lots of available substrates for ATP, so we're putting the extra stuff into triglyceride. So it's an indicator that we're meeting the demands. So we're going to start storing. So that would be an example of stimulatory to the center that tells you you're full. We've got some weird ones, some ones you wouldn't necessarily think of, but typically, if we get into extreme heat, it actually is going to signal your hypothalamus to tell you this is not a good time to eat. Eating actually causes an increase in body temperature because of the utilization of ATP and the requirement for ATP for digestion. So the extreme heat situation is also going to be stimulatory to the center that tells you you're full. And so the opposite to that one would then be extreme cold is going to be stimulatory to the hunger center. So generally speaking, in those types of environments, you'll be stimulated to either be full in the case of heat or to eat in the case of cold. So these are not insurmountable signals, right? You can overcome these signals. You can certainly eat when you're hot and you can not eat when you're cold, but these are just sort of reflex mechanisms. These are signals to these parts of the hypothalamus and what they respond to. Now, you can also appreciate that there's some mechanical stimuli. Stretch receptors, particularly in the stomach, but in the small intestine too, they would also stimulate signals to say, you don't need to eat more right now. We're already stretched. We're already full of stuff, right? So that makes sense, I think, that it would be a signal to say that you're full. Now, what do you think insulin would do? If insulin is responsible for lowering blood glucose levels, what would the signal that insulin might have on the hypothalamus? Stimulate that you're full or stimulate that you're hungry?
Full.
Yeah, because you should be able to tell because of where I drew the arrow, right? So insulin is responsible for lowering blood glucose levels. The reason we would lower blood glucose levels is because otherwise the blood glucose levels were high. And if blood glucose levels are high, it means that we're meeting demand, right? Typically. So this would be another influence, another way for insulin to influence blood glucose levels, right? By saying you're full, stop introducing new glucose to the system. We're already got plenty. So you could make an argument that glucagon might stimulate the hunger side of the hypothalamus, as an example of another influence. But so this is just a way that the hunger centers versus sated centers of the hypothalamus can be influenced, right? The obvious one is blood glucose, existing blood glucose levels. We've got hormones like ghrelin and leptin that directly influence the hypothalamic hunger reflex or hunger centers. So, they're pretty easy to explain as far as that goes. When we look at thermoregulation, these things will come into play a little bit there as well. So this is sort of a tie-in to the thermoregulation topic as far as that goes. So regulation of metabolism, essentially the regulation of eating, is going to relay through the hypothalamus. A lot of the mechanisms that we're talking about in this example here are autonomic in response. Okay, we're going to